Hi everyone. We've got another type of reproduction to talk about. It's cloning. You've probably heard a lot about it. But do you really know all the details? There's sometimes a lot of myth surrounding clones, lots of miscommunication and different ideas that really are not true. So let's get to the bottom of this cloning stuff. First of all, there's a few things to understand with cloning, that there is reproductive cloning, that's one type. There's therapeutic cloning, and then we also need to know what stem cells are. So let's start with the picture in our bottom corner of our screen. That's your first baby picture. That's a blob of cells after fertilization that are probably traveling through the fallopian tubes or oviducts, and they're going through mitosis. That is a zygote, and those are what we call stem cells. These are cells that are what we call undifferentiated, that they have DNA that's not really turned off or on, but open to become whatever. In fact, this is at the point where that zygote could divide into two masses of cells and grow into identical twins. That's how open they are to becoming whatever at that moment. In fact, in some animals, that happens a lot. For instance, in a jellyfish, when a zygote is created, they actually divide, or what we call bud off, into eights. So that would be octuplets, right? And so these are stem cells. Stem cells can grow into whatever. They're open to whatever. Their DNA, their genes have not been turned on or off, and all of them are working. They're non-specialized cells. Now, the best ones to work with are the zygote form right there. But we do have some stem cells in our own bodies. For instance, in our bone marrow, we have stem cells who are creating our red and white blood cells as we speak. So that's some examples there. Let's look at some reproductive cloning. Okay, after we're done here, we should be able to describe the difference between reproductive and therapeutic cloning, the importance of stem cells, and a process called somatic cell nuclear transfer. We're going to learn a little technology here as well. So starting off with reproductive cloning. Oh my goodness. Well, basically, this is asexual reproduction. And as you know, it happens all the time in nature, such as bacteria cloning themselves through binary fission, double their DNA and divide in two and create two daughter cells that are genetically identical to each other. Happens all the time. A bud growing off of a hydra and then falling off and then growing to be its own individual that's genetically identical to the parent. A piece of starfish falls off and that piece grows into a new individual. That's fragmentation. And again, it's genetically identical to the parent. And so if you see a pattern here with cloning, it creates genetically identical individuals to the original individual. Plants also can reproduce by cloning or asexual reproduction by growing new roots, shoots, or runners. And just mitosis itself is a form of cloning. When cells in your own body, these are called somatic cells or regular body cells, need to replace themselves, like your skin cells, replacing themselves. Usually that's about a cycle of every 30 to 40 days. Or your stomach cells are on a cycle of mitosis every 2 weeks or so. So asexual reproduction happens all the time. Cloning happens all the time in nature. But this is where people get kind of mixed up with cloning. They get these ideas in their heads, maybe from watching Star Wars, where you could make a clone army. Well, that's not quite the way it works. Do you know who this is? This is Dolly. She is famous. The famous sheep who is actually a clone. We never hear about who she was cloned from or what that sheep's name was, but Dolly made the news in the 1990s. So we'll get to Dolly in a moment. Here's some more cloning. When identical twins are created, that's by cloning. The individual zygote there splits into two, and we get two identical individuals. Also, scientists often can pluck off cells when they're experimenting with other organisms. They can pluck off cells off of a zygote and they can grow them into other individuals as well, identical twins in that way. And this has been going on for a long time, since the 50s and 60s, and has been used in cattle breeding for quite a long time. That's called embryo splitting. It's actually, to me, it should be more like zygote splitting, but the regular human being in the world calls them embryos, but really they're zygotes at that point. So let's talk about Dolly and nuclear transfer. How do you actually make a clone in a lab yourself, where you're not just like splitting embryos or splitting zygotes? How do you actually do that? Well, it's by nuclear transfer or somatic cell nuclear transfer. Again, a somatic cell is a regular body cell. I'm going to need to switch to another screen. Okay, so now I'm at this screen and I want you to picture a cell here. This is a cell from an organism that we want to clone. Just a regular body cell, and remember, regular body cells or somatic cells contain a nucleus. Not that it's always right there in the center of the cell, but that's often where we put it. Remember, in that nucleus, that's where your DNA is. Your DNA has all the genes to make all the proteins in your body, and then some. And a human has 46 chromosomes with the DNA all wrapped up. I'm not sure how many chromosomes sheep have, but different organisms will have different numbers. But a regular somatic body cell will have 46 chromosomes, at least in a human. So this is the organism we want to clone. So what we need to do in order to clone it is to get that nucleus with its precious DNA that contains the instructions to make an individual's proteins. We need to get that out. So there's these machines that have these really fine, teeny tiny little needles that they can stick into that cell and suck that nucleus right out of there. Then what we need to do is take that and put it into a donor egg. So that's exactly what we're going to do here. We're going to take that nucleus and we're going to transfer it over to here. So we need an organism of the same species to donate an egg, and we need to remove the nucleus out of that egg because we don't want to clone the donor. We want to clone whatever it is we want to clone here. So this egg, notice, has no nucleus in it, but it has plenty of cytoplasm, plenty of organelles, and all the stuff that a growing individual would need, but it just is missing the nucleus and DNA, and that's going to come from what we want to clone. So this is going to go in here. And again, there's machines and needles and such that can puncture in there and stick that right in there. Okay, so there's that DNA. So now we have made a full transfer. So that's where we're getting a somatic cell nuclear transfer. I'm transferring what I want to clone, its nucleus with its DNA, into a donor egg. So notice this egg now has a full set of chromosomes. Has all 46 chromosomes if we're cloning humans. Again, I'm not sure how many chromosomes sheep have. But since it has all of the chromosomes that it would need for that individual, what we're not going to need is fertilization from a sperm. In fact, we don't want it to be fertilized now because then it would be too much DNA. But we need to simulate somehow the act of fertilization, which can activate that egg to go into mitosis and become a zygote. That's what we need to do. And we can't let a sperm do it. Normally they think a sperm helps with that. So what they use are chemicals and electricity to activate that egg to go into mitosis, start dividing, and create a zygote. This is usually done in a petri dish. And if it works, then they'll have a zygote growing into a new individual in that petri dish. So what they would need to do after that is, if it worked, then they would implant that zygote into some surrogate mother. A surrogate mother is someone who volunteers to carry that baby, but it has to be inserted into the mother's uterus, and hopefully it implants. So often they'll do more than one of these at a time, because it's not always great odds that implantation will occur. So they might do a few at once in hopes that one will grab onto that uterine lining and implant itself. So this is also where sometimes it is successful and more than one grab on at a time. And this is where we got, if you remember, I think it's in the 90s, the Octomom, where she was going through a similar process called in vitro fertilization, where they fertilize eggs with sperm in a test tube or in a petri dish, and they let them become zygotes, and then they implant them into the uterus. And in those cases, she had many implant. But with Dolly, it seems that only one zygote took hold. And so then we got Dolly. So let's take a look at that. We have our we have our egg and the discarded nucleus from that. So then the egg is empty. Then we have our desired nucleus with the desired DNA and the recipe for the proteins to make the individual that we want. And we put that into the donor egg, zap it with chemicals and electricity to make it undergo mitosis, and then it forms a zygote, and then hopefully we will get a clone. But we're going to have to implant it into a donor mother and hope that it actually does implant, and that mother, that surrogate mother I should say, will then carry it as a baby to full term and deliver it. So your clone is going to be always younger than you, if you haven't figured that out yet. So here's Dolly, and that describes this and has her picture right there. Made Time magazine. This actually was done with other smaller creatures, but when it was done with a big sheep, that made the news. That was popular at the time. Again, we don't know who the mother was, the surrogate mother, and we don't know who the first individual sheep was, but we do know the clone was Dolly. So here's a picture of that happening, where this is holding the cell and this needle here is either plucking out the nucleus or putting one in. So here's someone actually looking into a specialized microscope in order to do that. I hope his hands are steady so he doesn't mess up. So that's what that looks like. So Dolly went on to be a sheep, live kind of a normal sheep life, and she got to have boyfriends and have babies of her own, the natural way, not the clone way, but the natural way. So Dolly had a little lamb, and oh, here she had a few more. This was 1998. But then the news hit. Dolly dies at the age of 6. She had to be put down by lethal injection in 2003. She had been suffering from lung cancer and crippling arthritis. Oh, I guess she was a Finn Dorset sheep. Most of them live to be about 12. So Dolly literally only got to live half of her life that like a normal Finn Dorset sheep would live. That sounds sad. Oh, let's see. The unnamed sheep from which Dolly was cloned had died several years prior to her creation. Oh, that explains that. So poor Dolly. And so, well, then the procedure becomes a little more popular. They get a little bit better at it, and they start doing this with some other organisms as well. For instance, this is Cece, carbon copy the cat, cloned in 2001. Like I said, the actual success rate can sometimes be bad. So it took like 87 tries for them to get this one cat. But can you imagine people who lose pets, how this could cause something in the future where they want to clone their pet because they miss them? But if you really look at it, when you have a clone like this, yes, the genetic material is identical, but there's other factors that affect the way you act and the way you look. So Cece the cat actually doesn't look exactly like what she was cloned from. This is what she was cloned from. And she looks like this, looks quite a bit different. This is the surrogate mom who took care of Cece, and probably the lady who had a lot of money to buy Cece. I don't know. Maybe she just adopted her. I shouldn't speak too soon. But you can see where there might be a problem with people expecting that when they clone something, a pet or let alone a human, that they would expect that individual to look and act exactly like the clone or like the original organism. But we know that that is actually not going to be true. They've done this with other organisms. These are some more clone kittens here. You can tell I like kittens, cats. This is a dog. I like dogs too. So Snuppy the puppy was a clone. This is the surrogate mom who carried and took care of Snuppy. And keep in mind, the United States, we're not the only ones who know how to do this. Other countries know how to do this as well. You'll have to look up the rules and regulations of all of this. So why would we choose to do this? Well, you know, we're humans. We got our big brains. We want to know how to do stuff. That's kind of goes without saying. But with farming, what if we could produce better animals or better organisms who could make more food for us? With therapeutic cloning, this is where you can take those early zygote cells and treat them with certain factors to make them grow into whatever you want. So we might be able to grow cells or tissues for maybe burn victims who need more skin that had it burnt off or destroyed, to create organs for people who need transplants, things like that, for people with nerve cord damage. Saving endangered animals. I don't know about that one. Bringing back extinct animals like the woolly mammoth. Oh my goodness. Or let's see, what else is extinct? The passenger pigeon, the quagga, which is an extinct type of zebra. I don't know. What do you think about that? To have a replica of your lost pet or loved one? What do you think about that? Most people, when it comes down to it, are okay, pretty okay with the therapeutic cloning, where you just take cells off of a zygote. That happens all the time in nature, and then grow them into what is needed. Plus, we're helping people. So most people don't have a lot of problems with that. Most people don't have a lot of problems with the farming and cloning of plants. They're plants after all. But what people tend to have a lot of problems with is the idea that we could bring back endangered animals or extinct animals, or let alone make a clone of a lost pet or a loved one. So what do you think about all of that? That's a good question, right? That might be a decision that you have to make someday. But before you do, you should understand some things. That it's not that easy. Like I said, there's not a great success rate, that it takes a lot of tries and a lot of zygotes or embryos die trying in order to make a successful clone. I'm guessing they get better and better at it with time. You need really good DNA for it. What if you brought back an individual that was an extinct organism? Where would you put it? Do you know how to take care of it? Will it destroy the ecosystem if we let it go? Because the ecosystem has already adapted to its absence. So those are all big questions. If it's the only one of their kind, who will teach them how to act and behave like they should? Right? And then another thing that I should mention, remember we said Dolly didn't live very long, right? It turns out that when you clone something, that you're not giving it fresh DNA from a sperm or an egg. You're giving it kind of old DNA that has probably undergone many rounds of mitosis from a body or a somatic cell. And it turns out that that ages a cell, and that's probably why clones tend to not live as long as something born from fertilization of an egg and a sperm the natural way. So that's why Dolly lived like half of a lifetime instead of a full lifetime that a Finn Dorset sheep should live. There's thoughts of bringing back the woolly mammoth. You may have heard of that. They're ligers. Do we even want to mention ligers? That's not a clone. But here's the slide I was looking for. Here's some of the problems that I've been discussing. The low success rate. One thing that we may not have mentioned is that, again, people who are in poverty might feel pressure to sell their eggs to become a surrogate. And so I can see some problems with people who are in poverty, uh, newborn clones dying, having a shorter lifespan. Some clones are born larger than normal, having their old DNA. The ends of the chromosomes, every time a cell goes through mitosis, the ends of the chromosomes kind of wear away a little bit. Those are called telomeres, the very ends. It's kind of like yarn or string. After you use it too much, or shoelaces, the ends get kind of frayed. That's kind of how chromosomes act in somatic body cells, and one of the things that causes aging of your cells. And so if we don't give it fresh DNA from a sperm or an egg, we can have some problems there. Again, the expectations of how clones should act or how they should look. And of course, we already have, if we clone humans, we already have 7 billion people or more on the planet. Do we contribute more to overpopulation? So some things to think about, and if you look up the SciShow, they have a lot of videos that have to do with cloning. You can just type it in, SciShow and cloning, and find all kinds of things. Why we age, stem cells, genetically modified humans or other organisms, and resurrection biology. There's a lot of things there. So this is uh, a very true meme here. Cloning results may vary. You never know what you're going to get. All right, so I hope that clears up some issues with cloning. I hope you understand it better and can discuss it with great intelligence now. Let me know if you have any questions.